study more characters. Sorry.
Yeah.
Yeah, so pea plant also has short life cycle, which means he could study more number of generations in a short period. Then he chose it because it produces large number of seeds. That means more number of progenies.
Yeah.
Then he chose it also because it can be easily crossed or self-pollinated. Then you asked us to read about dominant and recessive, and you asked us to
Characters.
read
Yes.
page 70 to 74 from the NCRT, and then
Yes.
you asked us to revise the DNA, chromosome, gene, allele, and mutation.
Yes. So
Yeah,
everyone did that. Everyone did the revision, your homework, people, because we are going to step into genetics. Now, see, genetics, before I start with this chapter, let me orient you to it, because whenever we start a new unit, I like to orient students to that unit and what is the significance of that particular type of study that we are going to do. So when we started reproduction, I did that. And in reproductive, like in the unit reproduction, we studied starting from bacteria to humans and even going to the health related issues with the human in chapter 4. Now, genetics sometimes students find is difficult because genetics is slightly different from other type of biological chapters or other type of biology that we study. But genetics is the most, like I believe, the most conceptual unit where you do not have to remember much, but you have to understand the concepts, you know. And this is the most interesting part of biology that I like, along with neuroscience, of course. So if you'll pay attention to this, if you'll miss classes, you may find a little disoriented or a little confused in the next classes, because we are going to read some laws, then we are going to practice them, we are going to find the answers. And in the next class only, I'll tell you the exceptions, that see, the law is not being followed everywhere because it's biology, right? We call it the science of exceptions, because we are studying life, and life does not have one single rule to follow. It always finds different things, right, in the system. So genetics is all about how any character, like genes, basically, which we talked about earlier as well, through gametes, which we have elaboratively studied in chapter 3, is inherited to the next generation. Okay, so what are the rules, what are the laws that are followed while any character is being inherited? Yes, Fatima. You have a question, Fatima?
I don't have any question.
Oh, okay, sorry. I thought you spoke something. So in this unit, I'll be heavily using concepts of DNA, chromosome, gene, allele, mutation, and that's why I asked you to go and read it. So let's begin with this only quickly, so that we also have a, so that I am assured that everyone is on the same page. Okay, so let's begin with DNA quickly. So you have to tell me, this is not what I'm not starting to teach. I'm just going to some backlog things. Okay, DNA, tell me, Fatima, what do we understand by DNA?
So that if me.
No, I'll go in a series, don't worry. Next is for Arpit, then Matthew, then Samira. It's just like your name is coming at the top of the list. Okay, so let's start with the DNA, the basic one. What do we know about DNA?
It's deoxyribonucleic acid.
Yes, so it's a molecule known as deoxyribonucleic acid. Yes, perfect. But what is it? What is deoxyribonucleic acid? Where is it situated? What does it do?
It consists of ribose, sugar, phosphate group, and nitrogenous bases.
Yes, right. And where is it present?
Inside the nucleus.
Yes, present in nucleus, basically, of eukaryotes, or in the nucleoid of the prokaryotes. So it's present in the nucleus, and what is it? This is the genetic material, correct? You always have read this word, genetic material, right? This is the genetic material of any organism. And what does it contain? What does it do? What is the function of DNA? Just to be there?
Whatever is inherited that is present inside the DNA.
Basically, so you mean to say that our DNA is the blueprint of everything that we are, right?
Yeah.
So how we look, how we behave, how we what diseases we are susceptible to, what infections we are susceptible to, what are our weaknesses, what are our strength in immune system, physiology, etc. Every information is present in the DNA. So DNA is the blueprint of living system. But still, what is the function of a DNA? What does DNA do?
Contains the genetic information.
Yeah, but what happens to that information? What does it do? What is the function? We understand that it is the genetic information. But what happens? We have the genetic information, then
It's inherited.
it is inherited. Let's say you have DNA inside you, right, Fatima? You are not like giving it to any further generation right now, let's say. But still, it must be doing something in your body also, or is it just a pushtani, khandani, zever to be inherited? Unless and until there, it will not be inherited. It will be kept in a box, closed. No,
No,
right? It's a very yeah.
it has genes which are responsible for proteins.
Yes, perfect. So DNA is actively in your body getting converted into RNA, right? Being transcribed. Remember, I told you the central dogma in biology?
Yeah.
DNA
Yes,
is
sir.
converted to RNA. This is called transcription. And then RNA forms proteins. This is called translation, right? And these proteins have all the function. Mostly everything in our body is done by enzymes, our proteins, all our different other proteins that we make in the body, structural proteins, which make our cells and other things, muscles, etc. Okay, so finally we have to reach to this level, and it comes through DNA. What was the next thing? Chromosome. Yes. Now, before going to chromosome, let me also talk about gene. What is a gene? Where is it? Quickly.
It's present in the DNA and it's the central unit of inheritance.
Yes. So
Part
gene.
of the DNA.
Perfect. So it is part or region of a DNA only, right? So which means the whole of the DNA is not whole of the DNA is not genes, but few patches are genes, right? There are
Yes,
other
sir.
parts of the DNA as well which are not genes. So what they do? What do they do? What are they called? NCRT calls them junk DNA because 5 years ago we did not knew a lot about them, or at least a decade ago we used to think that whatever is gene is the important part of the DNA. The rest is the junk DNA. And you know what is the proportion of genes in our DNA? You will be very, very surprised to know that. How much of the DNA is genes? You know that?
Sir,
Only
less than
1%.
Less than 1%. So still let me take it as 1%. So only 1% of our DNA is active functional genes. You know how many genes human have? Number.
So
Around 28,000. Okay, genes we have. And you know how many genes does a butterfly have? Around 32,000. Does this mean butterflies are more complex than humans? They are more evolved? They are better animals? Tell me, a butterfly has more genes, more protein coding genes than humans. And this was a very, very disappointing thing. United States started this human genome project.
yeah,
The
we
full
just
genome.
studied about it.
Yes, the full. We will study about this in this unit in genetics. The full genome of human have been sequenced. Okay, and when it was starting, everyone thought or hypothesized that when we will finish it, we will find somewhere around 1 lakh to 1.5 lakh genes in humans, because humans are very, very complex organisms and we are the most evolved. We already have had sequenced other lower organisms like yeast and like bacterias and like some lower invertebrates. So we thought that we must be having around 1 lakh to 1.5 lakh genes, but it came out to be around 28,000.
So
So
this
we,
is per cell or per body?
like the body is repetition of cells only. No, this is about like one haploid set of the genome. Whatever we have in like 23 chromosomes, the same thing we have in other 23 chromosomes that comes from father, right?
Yeah.
Yeah, understand. So these are the 28,000 genes, and we were very like, we thought that if humans can manage to be humans with 28,000 genes, and if butterflies still are butterflies with more than 28,000 genes, then just the number of genes do not decide how complex an organism is going to be. Yes or no?
Yes, sir.
Which means the 99% of our DNA is not coding for proteins. It is not making RNA and protein. So what it is doing? So we used to think simply nothing. But now with advanced studies, we are understanding, no, that's not the right answer. 99% of something cannot be just a waste thing. Yeah.
Yeah.
So there are regions of those DNA which affect this 1%, which are present. So this 99% of the part is responsible for the stability of this 1% and to keep human like stable as a species without mutating very fast or changing into things. There are enhancer reasons. There are all sorts of reasons that you will study if you pursue genetics in your higher studies, or if you become doctor, you will probably also study this in your medical. But just know that gene is the functional part of the DNA that codes for proteins. Okay.
So genes are also present in pairs.
Sorry.
Genes are also present in pairs.
In pairs.
Yeah.
Yeah, they have to be present in pairs, no? Because whatever, so chromosome number 1 from mother, let's say it has 100 genes. So chromosome number 1 from father will be the same chromosome, right? Having the same 100 genes. So in a diploid cell, they are always in pair, correct?
Yeah.
Yeah. So this is where we come to chromosomes now. Now you already know what will be chromosomes. It is condensed
Sir, it is.
form. Yeah, Arpit.
Sir, chromosomes are the thread-like structures which are present in the nucleus of the cell. Sir, each chromosome is like made up of DNA coiled several times, and sir, each cell generally contains 23 pairs of chromosomes.
Human cell, each haploid human cell, okay, contains 23, and diploid contains 23 pair. So when you say each cell contains 23 chromosomes or 23 pair of chromosomes, you have to specify that that cell is a human cell. Each Drosophila cell or a fruit fly cell contains only 4 chromosomes or 4 pair of chromosomes. Okay. So every organism has different set of their haploid genome. Remember I told you? Every organism does not have 23 pairs like human. Is it clear?
Yes, sir.
So chromosome is basically supercoiled DNA, right? And if it is DNA only, it also has the gene. So chromosome is supercoiled DNA wrapped around what? Proteins. And we call these proteins as histone proteins, around which DNA is coiled. And this has been asked in NEET, since it is not, even though it is not in NCERT, but you should know histones are positively charged proteins. And why do you think it's positive charged?
Because DNA is negative charged, and to
Yes.
for
Yes.
the DNA to coil around the histone, we need 2 opposite charges.
Yes, right. It's easy to attract 2 opposite charges simply using the law of physics. So if DNA is a negatively charged polymer, then it can be easily wrapped around a positively charged protein, which is called histone protein. Okay. So chromosome is clear, and inside the cell, like Arpit correctly said, these chromosomes look like threads, like these. So this is how they look inside the cell. They may be of varying lengths, some long, some short, but they look like threads because they are very supercoiled forms of DNA. And what was apart from that? Yes, allele. What do you think allele is?
It's a contrasting form of a gene.
Same gene.
Yes, right. So alleles are alternate versions, right? Alternate versions of the same gene. An allele can be 2, 3, or more. It's not like so that there will be only 2 alleles for each gene. There can be more than 2 alleles. Okay. So alternate versions of the same gene, they are mostly more than 2, like 2 or more than 2. So for example, capital T is a dominant form of allele which gives rise to tallness, and small t is a recessive form of the allele which gives rise to dwarfness, like we studied above. But both these two are alleles for the gene that controls height, right? And height is a character. Then these two becomes traits. Is it clear, everyone? All the words are clear to you?
Yes, sir.
These become traits. Okay, last is mutations. What are mutations? Any idea about mutations?
Sudden change.
Evolution.
Evolution is not mutation. Evolution and mutation are 2 different things.
Change in the genes. Sudden change in the genes.
Sudden change in the genes, which means, but any sudden change will be mutation. Any change will be considered mutation if it is a change in the gene. Let's say because of UV radiation, my skin cells got some mutation, got some change.
To protect themselves.
But will I say that that is a mutation? No, I'm saying, Arpit, that my hand was exposed to something, UV radiation, for example, and some cells in my skin of the hand got their DNA changed. Now, is it a mutation? Fatima?
So can you repeat what you said, sir? My net just lagged.
Yeah, I asked that any change, for example, if my hand was exposed to UV radiation and certain cells in my hand got their DNA changed, got a break in the DNA or got the DNA changed, will it be called a mutation? Like, will any change in the DNA be called a mutation, or is there any criteria which we have to follow to call something a mutation?
The codons being changed.
Yeah, codons. Yeah, DNA changes, codon changes. That's true. But any change will be a mutation. For that matter, we do gene therapy where we change the genome. Is it a mutation? No? Then how will we say something is a mutation? Mutation happens in genes, okay? And the thing which should be there, the criteria is that mutations are sudden, they are random, they can happen anywhere, but they are inheritable. They are passed on to the next generation. Do you understand, Fatima? If there is any change in your DNA which is not being passed on to the next generation, that is not mutation. Okay? So mutations pass on to the next generation. Okay? So write down, mutations are sudden, random, and inheritable changes. Sudden, random, and inheritable changes.
So, sir, no mutation cannot means like every mutation has to be inherited.
Yes. Mutations have to be inherited. Otherwise, if it is not inheritable, it is not a mutation. It is just a change due to some environmental factors which is creating the change. For example, let me give you an example. Any change that we acquire during our life, for example, in features, for let's say we started doing some therapies, you know. So any therapy which started making a human fair, more like, you know, these days we get tattooed. So when we get our skin tattooed, we have a different, we have a change in our skin now, right? Or if someone gets his or her hand cut, let's say I'm doing an experiment with mouse and I cut the tail of the mouse. Now that is a change in that mouse appearance. It is a tailless mouse. So when that mouse will give rise to another next generation, will the next generation be tailless?
Yes.
No. The next generation will have tail because the gene for the tail is still there in the gametes. It's just that that was not a mutation. That was a physical change that happened in that mouse. It was an acquired change. So changes are of two types, inherited and acquired. If someone is intelligent, then is the next generation going to be intelligent for sure? If someone knows Bengali, will the next generation know Bengali
No.
by birth? No, because all these changes are learned changes. They are acquired changes. That has nothing to do with, yes, please.
So what was the mutations in COVID virus? And like people used to say, na, it mutated and all.
Yeah, we'll come to COVID virus, and like when we'll do mutation. So I have kept that part for there. Okay, Arpit, can you just hold on for some
Yes,
time?
sir, no issues.
Yeah.
Yes,
Yeah.
sir, yes.
Okay. Cool. Thank you. Was Matthew saying something? I heard his voice. Are you asking something, Matthew?
No, sir, I didn't ask anything.
Okay. So yes, Fatima. So we were discussing that a tailless mice whose tail has been cut. If you keep cutting the tail, now I'm asking you a different question. If I do an experiment where every time I mate two mice, and when the generation comes up, I cut their tail. Then again I make them self breed. Then again a generation comes, I cut their tail. So if I keep doing this for, let's say, 100 generation, will after 100 generation I will get tailless mice?
Yeah.
No, I will never get a tailless mice, because every time, does not matter how many times I do a physical aberration to that mouse, the gene, there is the gene to develop tail still there in that genome. So every time the gametes are passed on, you know, I'm cutting the tail. Whatever I'm doing it after the birth. No, you have to think it like that. After birth, they had tails, so I cut the tails. But they already have gametes. They will form gametes which will have that gene, because, you know, they inherited it from their father. Only way to get a tailless mice is go to the genome, do a gene editing, make that gene that gives rise to tail inactive, or cut it out of the genome, which we call as gene editing. Only then we will get tailless mice. Is it clear? Do you understand?
Yes, sir.
Yeah. So does not matter how many generations you keep cutting the tail, they will always give rise to tailed mouse. Okay. So mutation is that sudden change. Let's say there were two mouse, both had tail, they were mated, and they had kids. And now some of their kids do not have tail. They are tailless, which means that in the gamete of that this progeny received, the gene for tail got mutated. You understand? Or had some issues. Yes? Is it clear, Fatima?
Yeah.
Okay.
Sir, can you like explain it once again, please?
Yes. So let's say we had a mouse which had tail, okay? And I crossed it with another mouse with tail, okay? In the next generation, I'm getting progenies. But let's say out of progenies, many progenies had tails, but one progeny was tailless. Okay, so this progeny was tailless. You understand?
Yeah.
Now, if only one progeny is tailless, it means that something went wrong with this progeny during development. That gene got faulty or did not work. If this mouse had mutation, let's say it has mutation M+ for tail, then all mouse still will be having tail, because the other parent will provide for that gene, right? There are 2 copies.
Yeah.
You understand? Either one way is that I take mutants, which are heterozygous. Mutation over wild type. If this is heterozygous for tailless mutation. So in this progeny, it must be mutation over mutation, right? So that's why it is tailless. Make sense?
Yes, sir.
Rest others as tails. Okay?
Yeah.
So mutation is clear, chromosome, alleles, gene, DNA, everything is clear. Now let's start with some hardcore genetics. Okay, everyone? Anyone who finds genetics difficult or boring, just please say, because I know how to make it interesting. There are some students in different other batches who found genetics boring, but now they are in love with genetics. Yes, Arpit, you find genetics boring?
Yes, sir, it's quite
Boring or difficult?
probably difficult.
Yeah, I thought like you should not find it boring. It should be difficult. But I understand. Okay, so let's make it easy now.
Sir, you know, I recently learned about DNA fingerprinting, and I thought it was so cool. Like that.
Cool, no?
That made me like, you know, it was so nice.
Yes, DNA fingerprinting is like just the fingerprint, but it's the DNA, so it, you know, it's foolproof. Okay, anyways, so we'll come to DNA fingerprinting also in genetics. I'll teach you that, but let's start with basics. Let's start with this one person who changed the course of biology forever, known as Mendel. Gregor Johann Mendel. So Mendel's laws of inheritance. You must have heard of this, right? In your junior classes, Mendel gave some laws in genetics, which are known as laws of inheritance, correct?
Yes.
What were the laws? Let's start with the first law.
Law of dominance.
Yes, perfect. First one is law of dominance. Now, what does this word means? Dominance. What is dominance? What is dominance? Matthew can tell. Liverpool dominated the game. What does it mean?
Yeah,
Matthew?
so basically, like, how do I say this in words? Uh, Taking control over something, or
Yes, they did not let the opposition, whoever was present with them, express themselves freely. They expressed their game. You know, they did whatever they wanted to do, right? That's called domination. If there is a fight between 2 countries, one dominates it. If there's a war, similarly, expression of characters is like a biochemical warfare. So law of dominance says that for every given gene, there are more than one alleles, right? So
yes,
when Mendel gave the law, Mendel knew and he thought that there are only 2 contrasting alleles for every gene. For example, the gene for tallness had a capital T and a small t. Now he did not know that these are genes, so he called them factors. So Mendel says that, and all these laws were given, like it came from Mendel's work, but they were not given as laws by Mendel himself. So you have to remember that. The status of laws were given by 3 scientists independently, mainly Hugo de Vries. We will talk about it later, but just know, let's start it simply as Mendel's law of inheritance. So law of dominance states, write down, law of dominance states that in a heterozygous condition, one allele or trait is expressed over the other allele or trait always. The other allele which is not expressed is known as recessive allele. Okay, and this is called dominant allele, and is called dominant allele, and the other one is called recessive allele. For example, the character for height has two alleles. One is capital T, giving rise to tall trait, and one is small t, right, giving rise to dwarf trait. Is it clear, everyone? Height
Yes,
in
sir.
pea plant.
But when Mendel when he did this experiment on pea plants, he didn't know what alleles are, right? He called them factors, right?
Yeah, he called them factors. Yes, he did not know what's happening, but now I'm telling you what was happening was this tall and dwarf. Fatima said in the beginning that we have alleles occur in pair, right?
Yes, sir.
Yes, sir.
Because chromosome occur in pair, right? Let's say these are the two chromosomes, chromosome 1 and 1 from mother and from father, let's say, and it has the gene for height here. So both will have the gene for height at the same location. Mother carried a type of the gene, a version of the gene that was giving rise to tall character, but father had a version of gene which was giving rise to short character. Now these two are different alleles of the same gene. Alleles are always present on different chromosomes. Is it clear, everyone?
Yes, sir.
That's why only one got expressed over the other when they were present together. So this was law of dominance. When Mendel did his experiments, he knew nothing about alleles, genes, or chromosomes. That came later. Mendel was very early in the 19th century. Okay.
Sir, so in codominance, what happened?
Yeah, we are coming to codominance. Okay. Let's first study law of dominance, no? Now, what Mendel did exactly, let's go through that. What Mendel did is he took a tall plant because he had to study characters, and all he knew was he took pea plant because pea has tall and short. So pea does not have an intermediate height like humans. In human, it was not making sense. And Mendel received a lot of backlash. His work was not recognized. No one wanted to believe him for a very long time because what he was talking was not making sense with respect to humans. Do you think that humans only are tall or short? There are two ranges in which human heights fall. No, right? It's not like
No.
either you will be 6 foot or 5 foot, nothing in between. But for pea plant, it was like that. Either peas were tall or short, nothing in between. So he took a tall pea plant and crossed it with a dwarf pea plant. So these were the parents. In the first generation, he saw that all the plants were tall plants. Now he thought, wait a minute. One parent was tall, another parent was dwarf. How come all the progenies in the next generation are tall? Where did this dwarf characteristics went? But he could not understand it at that time. But now we know that. Okay, but before that, let me take the story forward. Then he decided to do one thing. He self-crossed F1. All were tall in F1, right? So when he crossed a tall with a short, he got all tall. He thought something is happening, something is not right. When he crossed two tall plants, so this was F1 tall crossed with another F1 tall. Now to his amazement, he found that now he got progenies which were tall and few of them were also dwarf. But again, wait a minute. None of the parent was dwarf, right? None of the F1 progeny was dwarf, right? So how come when there was a dwarf parent in the next generation, there was no dwarf progeny? When there were both tall parents in the next generation, there was a dwarf progeny. He was mind boggled. And he started thinking that just by sheer biological explanation, I will not be able to explain it. So he went back to mathematics. You know who was Mendel basically? He's one of my favorite characters from science history. Mendel was not a biologist. Mendel was a physicist. His formal education was in physics and he loves mathematics. And as a profession,
Yeah,
he was
sir,
a
we discussed this in the last class actually.
we discussed, yeah. And his
Yeah.
profession was a priest. So one reason that no one believed Mendel in the beginning was they thought that, oh, he is some physics person talking about some mathematics thing and applying it to biology. He does not understand biology. And that was true. Mendel did not understood biology by that time to an extent what other biologists know about cell and all these things. But he thought, okay, if there is a tall plant and there is a dwarf plant, mathematically, both of them are fusing, then some combinations must have been dwarf, unless this dwarf character, whenever it is present with tall, it gets masked, right? So to make a plant tall, there are two possibilities. Either it is having allele capital T, which is a pure tall. You understand, everyone? Or it is having capital T and small t, which is called hybrid tall. You understand? In both
Yes.
of these conditions, the plant is tall. So he realized that this tall plant was a pure line tall with which I started. And this dwarf plant was a pure line dwarf. And because they both were crossed, so the parents, what different kind of gametes they can form? Let's make a Punnett square here to understand it. This is called a Punnett square. Now parent one, I'm writing this side, it can either form a capital T or a capital T, right? And this parent can form two gametes, one having small t, another having the same small t. Now all the progenies will be capital T, small t. Do you understand? Is it clear, everyone?
Yes, sir.
Am I audible, people?
Yes, sir.
Yes, Matthew, do you understand it, or is there any confusion?
Yes, sir, I understood it.
Okay, so he realized plant was dwarf because for all of them the genetics was like this. The factors, these were the two factors that they got, and he realized that one of these factor is recessive. It never expresses in the presence of a dominant factor. You understand? This is how he figured it out. And when I crossed them, then in the next generation, so when I was crossing, I'm crossing a capital T small t with another capital T small t, right? And again, going through the same logic, let's see how many gametes both the parents can make. Now this time, both of the parents can make two different gametes, capital T small t and capital T small t, yes? And four possible progenies could be capital T, capital T, capital T small t, capital T small t, and small t small t. You all can figure out how I'm doing this, right? It's very simple. And he realized that three of them, these three were tall, right? And one was dwarf. Is it clear? So he found that there was a ratio of 3:1 for tall and dwarf. How he figured out the ratio? Suppose he got 400 progenies, he counted each, and then he got 400 seeds. Then he planted all those 400 seeds, and he kept doing it for years. He did all these experiments for 7 years. It took him 7 years to realize all these laws and everything that he wrote. Suppose he got 400 total seeds from the cross of the two plants. Then he sowed all of those seeds and saw how many of them are tall and how many of them are short. So he noted it down. And he realized that every time the ratio used to be 3:1, 3 tall and 1 dwarf, which means out of 400, 300 will be tall and around 100 will be dwarf. You understand? Because every time it follows this law. And dwarf can only express itself, a recessive gene can only express itself when it is in a homozygous condition. So this was another thing he found. So write down, a recessive allele can only express in a homozygous state. Okay, So in the F2 generation, he realized that you get that. In F1, everyone is heterozygous, so everyone is tall. In F2, you get one homozygous dwarf. So even you are crossing two tall plants, you get a dwarf progeny. Is it clear? So this is known as the genotypic ratio. What it is called? Sorry. No. This is known as phenotypic ratio. Phenotypic means what we can see. The phenology is the study of visible characters. So this is called phenotypic ratio. What we see. Because in these three tall plants, one is a different kind of tall. Can you see that? One is pure tall with respect to genetics, but visually both of them will be tall. And two are hybrid tall. So then he figured out that there is another ratio that we have to study, which is called genotypic ratio. And the genotypic ratio will be for different genotypes. Sorry. Capital T is to capital T small t is to small t small t. And what was the ratio? 1
is to 2
is to 2 is to 1. 1 is this, 2 are these, and 1 is this. Is it clear? The genotypic and the phenotypic ratio is clear, everyone?
Yes.
Everyone? Any doubt? Please note this down, starting from here, and then let me know. Someone has a question? Fatima, what do you want me to do?
No, so I was saying that can you keep the test next week, because tomorrow I have to go somewhere, so I won't be able to attend the class.
You won't be able to attend the class at all.
Yeah.
Okay. So you're also going to lose some genetic concepts. Actually, Actually, the point is that tests are already delayed because I wanted it to happen last week, but since a lot of you were going through examinations or were not attending classes regularly, so instead I thought of finishing chapter 4. So it has to get done because there is a schedule. If one test gets delayed, then everything we have planned in line gets delayed by 1 week. So that's the problem. Yeah. I'll provide you with the test paper. So you will miss the test, but you can do one thing: sit honestly and solve it by your own, and send me your answers on the WhatsApp group.
I'll do that.
Yeah. Everyone noted? Shall I move forward?
So tomorrow, what are you going to do?
Tomorrow?
Yeah,
whatever I'll finish today, from there onwards. Probably we'll try to finish law of dominance, its exceptions, because you talked about codominance. We'll try to do its exceptions today, and tomorrow we'll start with second law and third law.
Okay.
Third law is a little more complex, so let's see. You come up with doubts if you have any. Okay.
Yeah.
If missing the classes is inevitable, then this is what we can do. Done? Arpit, Matthews, Myra?
Yes.
Mehraj?
No, sir.
No? Okay. Take your time. It was very interesting when I read about Mendel and his work. He actually did some brilliance. He produced some genius of a work which stayed unappreciated for a very long time. Very, very long time. Like for decades, no one paid attention to what Mendel did. Everyone thought that he was some crackpot who just wanted to sow peas and write down something. And when he said that, I found about that there are factors, then everyone thought that, oh, what factors you're talking about? He said that there are two discrete factors for every gene that do not blend. Then people are like, ha 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 ha. What are you talking about? We have blending of characters every time in humans. Look at the skin color, look at the height of humans. And they did not appreciate his work because it was very, very fundamental. Then 3 scientists independently at like the beginning of the 20th century rediscovered his work, studied it, and realized he was a genius. And they gave the status of laws. So the laws that we study today are given by some other scientists based on Mendel's work. So they are called Mendel's law of genetics because Mendel did all the hard work. He, all the sweat and blood was his contribution. So of course, it has to be his laws. But when he was alive, yes, Arpit, done?
Yes, sir.
Very good. So you understood law of dominance, that in the presence of in a heterozygous condition. So there are 3 conditions that we can see. One is this. This is a homozygous condition and it is called a pure line tall or a homozygous tall. Okay, everyone, write down these notations. I will be giving you questions with this kind of terminologies. Okay, it is also called pure line tall homozygous. There is one more term. Okay, when I remember it, I'll tell you. Another is this. This is called hybrid tall. Okay, or heterozygous tall. And the third one is small t and small t, which is possible. And this is always dwarf. And dwarf or short has to be pure line because it's a recessive. So it's called homozygous recessive. These are homozygous dominant, heterozygous dominant. Okay, this is clear.
Yes, sir.
So they also tell you what is a recessive gene. A recessive gene is which is not expressed in the F1 generation. According to this flowchart, if you have in parents, one is dwarf, one is tall, the recessive gene, which is dwarf, does not crop up, does not show in the F1 generation, then again crops up in the F2 generation. Yes, that's the recessive gene. But and boards or entrances are not going to ask you very simple questions like this. Okay, so let's do one question and let's see. You tell me the genotypic and phenotypic ratio. Okay, shall we do a question?
Yes, sir.
Write down. A heterozygous tall plant was crossed with a dwarf plant. A dwarf plant. Find out A, the genotypic ratio, the phenotypic ratio in next generation. In next generation. Okay, let's start with this. Do it quickly. Let me see. Whenever and whoever is done can write in the chat the answer, the ratio, both GR and PR. So there's the same rule. Whenever you are given a phenotype, find out the genotype of the parents, then make the Punnett square with different types of gametes that are possible, then do the cross and tell me the ratio. I hope everyone is doing it. I received Arpit's answer, but what is it, Arpit? Genotypic ratio or phenotypic ratio? At least write GR or PR. You have to find both, okay? Matthew, you just made a very tiny mistake, silly mistake, which will cost you all the marks. See, there is no step marking here, though it involves some concepts of ratios and mathematics. In biology, you do not have step markings for solutions or processes. One small mistake at the top and 0 at the down of the pyramid. Matthew, would like to consider? Are you redoing it, Matthew? Just look at your solution, okay?
Yeah, I am doing it again.
Others, Fatima, Samira. Samira, you're there?
Yes, sir.
Oh, good to hear your voice. You're not speaking. So in the beginning, I will allow you time, if you're a little slow, but all I want from you is right answer. With time, I will expect it to be faster and right. Right now, I expect it to be right, if not faster, because you will not get this much time in your boards or entrances to spend on one question. But this is what I'll prepare you for, don't worry. Arpit, what is this ratio? Is it genotypic or phenotypic? You have not answered.
Sir, I sent, I guess. Sir,
Okay. I'm waiting for Samara's answer and then will discuss. Good job, Matthew. Okay. Good. Let's start. So, Matthew, would you like to help me with this?
Yeah, sure, why not?
sir,
I'll follow your
please
lead, okay?
consider the second answer.
Yeah, I've considered it. And we'll discuss why it is not right. Okay, so let's start with two parents. So let's clearly write parent A is heterozygous tall, right, Matthew?
Yes, sir. Yeah.
So what will be the genotype of parent A? Tell me.
Capital T, small t.
Perfect. And it's crossed with a dwarf plant, which is parent B. So what is
Small
parent
t,
B?
small t.
Small t, small t. So we are making a cross of this, right? If you figured out this, half of the battle is done. Now you have to figure out what different gametes can it make. For that, you can make a Punnett square because it's just single gene. So it will have two alleles. So on this side, I will write gametes of A and gametes of B. So for A, it is going to be what?
Capital T on one bone. Okay. small t on the other,
Okay,
and small t and small t again.
this is the mistake you made in the first attempt. Yes, right. So this will be
Yeah.
capital T small t. This will also be capital T small t, one coming from here and the other coming from here. Yeah. And then these two will be small t and small t small t, correct?
Yeah.
You see this? So talking about the phenotypic ratio first. So following law of dominance, there can only be two phenotypes, either tall or short or dwarf. So how many of them are tall?
2.
The first two, these two are tall, right, everyone? Phenotypic
Yeah.
ratio will be 2:2 because two are tall and the other two are dwarf. And talking about the genotypic ratio, there are two genotypes possible by this cross, either this one or this one, right? There is no homozygous tall here. Is there anyone homozygous tall?
No.
And these two are capital T small t, and these two are small t small t. So both genotypic and phenotypic ratio is 2:2. Is it clear? In the
Yes, sir.
F1. So if this is the parent, if you consider this as the parent generation, then in the F1, this is the ratio, or in the next generation, this is the ratio, right? So it's always important to figure out the genotype of the parents to say what will be the ratio in the F1 or F2. NCERT wants you to remember that in the F1, okay, people just 1 second. Just excuse me for 1 second, okay, I have to take this. Okay, am I audible and visible, everyone? As I can see, we have Vafia among us. Vafia, can you hear me? Am I audible to you, Vafia? If you're speaking, you're not audible to me, okay? But I just want you to like be in the class. And yes, Vafia, you're audible to me. Am I audible to you?
Yes, sir.
Yes. So Vafia, you have joined a little early, but it's always good. Some knowledge is always good. So stay in the class. We are discussing Mendel's law of inheritance, and then when this class will end, I'll talk to you. Okay. By that time, you can just take this class as your trial. Okay.
Okay, sir.
Cool. Okay. Fine. Thank you so much. Okay. So everyone back. So I was telling you that so NCERT tells you that in the first generation there is some specific ratio which is like all are tall and all are heterozygous. Then in the F2 ratio, in the F2 generation, the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. And I have seen students mugging it up by heart and going on exam, and whatever they ask in question, if they read F2, they just write these two ratios and come back home happily that we gave the right answer, which happens to be the wrong one, and they lose marks. So you do not have to just blindly remember ratios. You have to realize here, when we started the cross, we took a pure line tall and a pure line dwarf. You understand? This is what Mendel also did. Then when we self-crossed F1, we got both heterozygous tall. So that's why this ratio came up, because the parents, now for the F2, this becomes the parents, right? Yes or no, everyone? Your grandparents are parents to your parents, right? But for you, your parents are parents, not your grandparents. Do you understand? Someone should say
Yes, sir.
yes. So always in exam, when you do these crosses, pay attention to what are they asking you to cross. what are the parents and what is their genotype, because we saw that the ratio changed and it is not either 3:1 or 1:2:1, but it is 2:2 for both in case where we crossed a heterozygous tall plant with a dwarf plant. Okay, everyone? Make sense?
Yes, sir.
Now tell me another thing. I'm just adding to this question. If in the same question, like there is part B of the same question, let's say, if in the same question, I am crossing a homozygous tall with a dwarf plant, what will be the genotypic and phenotypic ratio in the next generation? Very quick.
Can you say the question once again?
Instead of homozygous tall plant, if I'm writing, sorry, instead of heterozygous tall plant, in the same question, if I change this word heterozygous and I write it as homozygous, then what will be the genotypic and phenotypic ratio in the next generation? Can you do it quickly?
Capital T, small t.
Everyone will be a hybrid tall, right?
Yeah.
That is the first, that is equivalent to what Mendel did, right? This one. Yes, a homozygous tall with a dwarf, right? So can we actually, through crossing a homozygous tall, so if here, if I'm crossing a homozygous tall with a dwarf, I get everyone as capital T, small t. So all will be hybrid tall, right?
Yes, sir.
Now just look at these two crosses, this one and this one. In this cross, one is common, right? This partner is common, which is called a recessive, homozygous recessive partner. Can you think that this type of a cross, because if we see something, if we see a dwarf character, we do not have to know its phenotype, its genotype. We already know that dwarf is a recessive character, so the genotype has to be homozygous recessive. Yes?
Yeah.
Everyone understands?
Yes, sir.
So this we know by just looking at the plant. This we cannot say anything about by just looking at the plant. If you look at a heterozygous tall plant, it will look the same as a homozygous tall plant, correct?
Yes.
So can we use this cross as a test to figure out what is the genotype of the tall plant? In the next generation, if I get 2 tall and 2 dwarf, I will say that the tall was heterozygous tall. And if in the next generation I got all the tall plants, then I can say that the tall was homozygous tall. Can I say that?
Please explain again, sir.
Yes, I will. The problem is that if we look at a dwarf plant, we can very easily say that what was the genotype. Small t, small t. No other combination will give dwarf. Yes? There are 2 alleles and it has to be homozygous for recessive to give a dwarf recessive character. Perfect. But for tall plants, there is a problem. We can look at a tall plant and can say that, oh, these 2 are tall plants, but we cannot be sure of their genotype, whether they are heterozygous tall or homozygous tall. Yes?
Yes, sir.
But the cross that you just solved right now, can this be used as a test cross? We take a tall plant whose genotype we do not know and we cross it with a dwarf plant. Dwarf plant will always be small t, small t. And if in the next generation I get half tall and half dwarf, then I can say that the tall parent was heterozygous tall. And if in the next generation I get all hybrid tall, no dwarf, then I can say that the parent was a homozygous tall. Can I do that? Do you understand, Arpit?
Sir.
No.
No.
Pay attention here. I have to find out the genotype for this tall plant. There is a plant which is tall. Okay.
So,
But I do not know the genotype. If I cross a tall plant with a dwarf plant, okay, you always know that what is going to be the genotype of a dwarf plant.
small t, small t.
Right? This part is clear to all of you.
Yes, sir.
We do not know what is the genotype of this tall. Now, what are the 2 possibilities?
Sir,
It can either be this or it can either be this, right? But just by looking at the plant, how do we say that? We cannot say that. So if I take any plant and let's say it was this one, okay, let's say the plant was this one. So when I will cross it with a dwarf plant, in the next generation, I will get this. On one side, it's capital T, on other side, it's small t. So I will get half of them tall. Sorry. I will get
everything tall.
all of them tall, right? So if I do this cross, if I cross a tall plant with a dwarf plant, and in the next generation, all of them are tall, then can I say that the tall father was homozygous?
Yes, sir.
Yes. Now do you understand?
Yes, sir.
And if I do this cross, so let's say the genotype was this, then in the next generation, automatically I will get half of them tall and the other half as dwarf. Correct? That's the difference. And if I get half of them dwarf by crossing a tall and a short plant, I know that the tall was heterozygous tall. And if I get all tall by crossing a tall and a dwarf plant, I can say that the tall was homozygous tall. Anyone still have any doubt?
No, sir, it's clear.
Fatima, you have some doubt?
So, sir, by seeing the F2 generation, right?
F1, let's say these are the parents. So, but just by looking at the next generation's phenotype, we can tell about the previous parent generation's genotype. Handy. Good thing, right?
Yeah.
So this type of cross is known as a test cross. I just taught you test cross without teaching you anything. We just did a cross, and you, just like every one of you, just like Mendel, realized, oh yes, it is a test cross. We can do it to test the genotype of a tall parent, a dominant allele. Yes, whether it is heterozygous or homozygous. Yes or no?
Yes.
So write down test cross. Let's define what is a test cross. Write down. It is a genetic cross between a homozygous recessive, recessive, which means small t, and a dominant phenotype, between a homozygous recessive and a dominant phenotype whose genotype is to be determined. Whose genotype is to be determined, whether it is heterozygous dominant or homozygous dominant. Whether it is heterozygous dominant or homozygous dominant. Is it clear?
Yes.
And also write down, if case 1, this is case 1. Case 1, if by crossing a tall, or not, let's not say tall, right? Let's say if by crossing a dominant and a recessive phenotype, we get all the progenies dominant of dominant phenotype. If by crossing, yes, if by crossing a dominant phenotype with a recessive phenotype, we get all the progenies with dominant phenotype. We get all the progenies with dominant phenotype. Then the dominant parent was homozygous. Yes. Then the dominant parent was homozygous. Is it clear?
Yes, sir.
And make this and just complete. Then you will then make case 2. Same statement. If we cross dominant phenotype with a recessive phenotype, and get equal proportion of dominant and recessive phenotypes in the next generation. 2:2, right? Equal proportions of dominant and recessive phenotypes in the next generation. Then the dominant parent was heterozygous. Is it clear, everyone? Clear?
Yes, sir.
Yes, sir.
Please make these two for the two cases, case 1 and case 2. And this is test cross. So you understand test cross? You understand law of dominance. You understand how ratios come up.
Yes, sir.
You understand how to solve any given question, right? Let me know when you're done. And Wafia, you also please keep noting these things down, okay?
Okay, sir.
Let me know, people, when you're done. I hope nothing is difficult or confusing till now. Everything is smooth, done. We all are on the same page, right?
Yes, sir.
Very good.
Excuse me, sir.
Yes, please.
Sir, I am getting a doubt again, sir. Sir, when we didn't knew that the tall plant is both capital T or that small, sir, how
Yeah.
then we can cross it like in the generations? How do we get the ratios then if we don't know the tall plant?
See, to cross is to pollinate a tall plant with a dwarf plant. That's all you have to do. That is what crossing means. When Mendel did the cross, he took a tall plant, and pea plant, you all know that they are cleistogamous, right?
Yes, sir.
If you do not do anything, they will do autogamy. Yes.
Yes,
So
sir.
he took pea plants, he cut the petal, he opened the flowers, he removed anther from one, from tall he removed the anther, let's see, and from the short he removed the stamens, sorry, the pistil. Then the only possibility was if there are two plants in close vicinity, then they will cross pollinate. Yes. And this could be done in pea. Once you cross pollinate, you cover that flour with a bag so that no other contamination is done. Then when the flour develops into a fruit, it will become pea pod. Yes. And the ovules will become pea seeds. Yes. Then Mendel got all those pea seeds and sowed them. And when you sow, let's say 10 seeds, and you see all 10 of them are tall, but you did a tall and a dwarf cross. Remember?
Yes, sir.
You do not have to know genotype for that. Genotype we are inferring. This is mathematics. We assume you all have done mathematics, no? Let's assume the value of x to be this, and then you figure out the equation. And if that equation fits with the standard, then it was right what you assumed. Otherwise, you will assume something else, right?
Yes, sir.
That's the beauty of mathematics. It's a very pure. It's called queen of science. Mathematics is called queen of science as a subject because it explains every other subject. Okay. So he just did physical crosses, and when he sowed all those 10 seeds or 20 seeds and all were tall, he went back to realize that the tall must be a homozygous tall. That's why this happened. So this he did on a piece of paper after he crossed two plants and got the progenies and saw that everyone is tall. Then only this is the possibility, no? If it was heterozygous, then this would have been the possibility. He would have got half of the plants tall and half of the plants dwarf in the next generation from the seeds.
Yes,
You understand, Arpit?
Got
it? Perfect. It's a beautiful doubt, actually. You know, there are many doubts that are beautiful. This is one of them. How did we figure it out when we didn't know the genotype? Yes, we did farming. Mendel did farming to figure out science, pea farming. Me and Matthew were discussing. Matthew suspected that probably he was selling the peas. Mendel Peas Corporation, MPC. Right, Matthew?
Yeah, it would have been a good business idea.
Of course, it would have been, right? If Matthew would have been there as an apprentice, he would have suggested this to Mendel. That you do all your science, you are generating this ton of peas, give it to me. Yeah, some side business is always good. Okay, everyone done? Shall we move forward?
Yes, sir.
Is the test cross easy? Now there is one more thing in the textbooks known as back cross. And this does not need any explanation. It means going back one generation and crossing. For example, anything, any parent you take, whatever is the parent's genotype, you got some progenies. If you are crossing the F1 progeny with any of the parent genotype, then it's called a back cross. Is it clear? Simple. If in this cross, I got this capital T small, so this is the progeny, right? Look here, this is the progeny, everyone.
Yes, sir.
If this is the parent generation, this is the progeny F1, right? If I cross this with the same, it is called self cross. If I cross this with any of the parents, either this or this, it is a back cross. Is it clear? Crossing it back with the parent's genotype. Is it clear or no?
Sir.
Yeah, so write down a back cross. A back cross is crossing the progeny with one of its parents. If the parents are dead, then something which is genetically similar to the parent will also do. And most of the time we do it for a hybrid organism. Of course, it has to be a hybrid, right? So write down back crossing is crossing of a hybrid offspring with one of its parents. Or another organism which is genetically similar to its parent, or another organism which is genetically similar to its parents. That's called back cross. Okay. So in a way, can I say that test cross is a special type of a back cross only?
Yes,
Can I
sir.
say that, or can I not say that? Think about it first. Is test cross all the time a back cross? Yes, it is, right? It is a special type of a back cross where we are choosing that parent which is homozygous recessive all the time. So all back crosses are not test crosses, but test crosses are back crosses. So you can write down. Let me write this down. A hybrid of offspring crossed to one of parents or individual having same genetic. So write down test cross is a special type of back cross only where we choose the homozygous recessive parent. The homozygous recessive parent. Is it clear, everyone? Test cross, back cross, law of dominance, genotypic ratio, phenotypic ratio. Everything is clear?
Yes, sir.
Okay, so now let's come to write down few more things about law of dominance. We are coming to exceptions of law of dominance. Write down law of dominance. Law of dominance is not a universally accepted law. Is not a universally accepted law, which means it has exceptions in biology. Mendel did not know about it. Now, this was later figured out that whenever there are two traits, Mendel said that one of the trait will always dominate and express itself in the presence of another trait in a heterozygous condition, right? This is what is the definition of law of dominance. But scientists realize over time that this is not holding true for all the traits in all life forms. So there was something called incomplete dominance. In some organisms, if there is a heterozygous condition between two alleles, one allele is not able to completely dominate itself. And because of that, there is a blending between the two alleles. You understand? That was called incomplete dominance. And it was found in the flower color. One example is, it is found in many organisms. I'm giving you the NCERT relevant example. Flower color of Snapdragon plant. There is a plant known as Snapdragon, and the flower color shows incomplete dominance. In this, what happens is, so the flower has two colors, has two alleles. So the gene for flower color. So there is a gene for flower color, let's say, having two alleles. allele 1 is, let's say, capital R, and this allele is responsible for red color, which is dominant, and allele 2 is small r. Is it clear? It has two alleles. Now, whenever it is capital R, which is a homozygous dominant condition, the color of the flower is red. Whenever there is small r, the color of the flower is white. Is it clear, everyone?
Yes, sir.
But whenever there is capital R and small r, what should have been the color of flower following laws of Mendel? Mendel's law of
Should have
dominance.
been red.
It should have been red, but instead,
It's
this
pink.
gives rise to pink flowers.
So like an
Now,
intermediate.
yes. So now we know that colors in any biological system has colors inbuilt. Like that color comes from pigments, right? Any color comes from pigment. Your eye color, your skin color, flower color, for that matter, they are all pigments. Yes or no? And pigments are proteins, right? And we know that proteins are formed from RNA, which are transcribed from DNA, which is a gene. Yes. Protein coding genes. Yes. So capital R allele must be giving rise to a protein, which is responsible for red color. And small r allele of the same gene is giving rise to another protein, which gives rise to white color. So white does not mean absence of any color. Absence of any color is transparent. White is a real color. You can see this background. It is white because it is color. Yes. So white color is also because of white pigments. Our eyes are white because of white pigments. Do you understand, everyone?
Yes, sir.
But when these two alleles are present together, one should have dominated the other, but instead both are producing some bit and getting blended. So when you mix, how many of you have painted as a kid or still like painting? Suppose you have no pink color in your acrylic set.
No, then you mix white and
Yeah.
red, or like if you have like a very light color and want to make it dark, you put like a black in it and try to make it dark.
Exactly, right? You can make pink from white and red. This is what nature is also doing, because both these alleles, no one is able to show dominance over the other. So the R is trying to make it red and the white is trying to make it white. And because of the combined effect, both of them, because of lack of any dominancy, it becomes pink. So this is called blending of trait, or blending of allele. Is it clear, everyone? Say yes or no.
Yes, sir.
So in this, the smaller protein produces more of white pigment.
Not more. It's not producing more. Maybe the reason could be there could be three reasons. Either this capital R is not producing red, or producing very less amount of red, which is not able to dominate over the white, or both of them are producing lower than they should have, so they are blending in to produce pink. But anyways, no one is completely dominant over the other. So there is an incomplete dominance and there are pink flowers, which is blending of the alleles. Is it clear, Fatima?
Yes, sir.
Yeah, there could be all permutation combination reasons. But this tells that if I do a cross, now in incomplete dominance, our concepts of test and back cross will not work now. Let's see why. So to begin with, if I have a red flower, now there are three phenotypes possible: red, white, and pink. Unlike tall and dwarf, right? There was no intermediate height.
Yeah.
Yes. So if I say that, let's do this question. Let me give you a question on this. Write down: a red Snapdragon flower crossed with a white. Start from the basic. Okay, these are the parent. What will be the genotypic and phenotypic ratio in F1? Quick, now this has to be fast. Capital R, small r, the first generation.
All of them will be pink.
Yes, right. All pink flowers. So no genotypic or phenotypic ratio. Till here, it still makes sense. In law of dominance, all were tall, like the parent. In here, the F1 generation is different from both the parents, right? Different from both the parents, both genotypically and phenotypically, correct?
Yes, sir.
So write down this statement, both genotypically and phenotypically. In normal law, by following law of dominance, the F1 progenies were different from the parents, but only genotypically. But phenotypically, it was similar to one parent, which was tall, right? The dominant parent. Here, it is different from both the parents, both genotypically and phenotypically. Now, my next question is, what happens when we self-cross F1? Tell me the genotypic and phenotypic ratio. Self-cross F1. This you will solve and tell me in the chat quickly. I have received one answer. Good, Fatima. Why? No. Oh, yeah, not good. Good was for solving it faster, but not good for solving it incorrect. Samara, I know that you are solving the cross correct, but let me give you a hint. You are not paying attention to the incomplete dominance, the blending part. Phenotypes will not be same for both homozygous and heterozygous this time. Sorry, Samira. Samira, please go back and cross check and tell me the answer again. Wafia, I'm expecting answers from you as well, because you're a part of the class, so you can start sending answers if you're understanding. You are understanding the concepts, Wafia? Is it clear to you?
Yes, sir.
Okay.
Sir, did you get my answer?
Yes, I did. I got your answer. You are rocking, Matthew. I think genetics is your forte, right?
I also sent.
Matthew's thinking, was I Mendel? Few centuries back. Okay, so I think Fatima will help me this time. Fatima, are you there?
Yes, sir.
Sir, did you
Let's
get
start.
my answer?
Yes, I got your answer. Do you want feedback or let's just solve it? You will understand, okay?
No feedback.
Thank you. Okay, great. Fatima, help me. So self-crossing F1, what will be the first parent? Genotype.
The first parent will be capital R small r, and the other parent will also be capital R small r.
Yes, Samara, very good.
Mth will be capital R comma small r and capital R comma small r.
Okay, so we are here. Say capital R, small r, and capital R, right?
Yeah, the
For
first
this.
progeny is capital R,
Mm-hmm.
then it is capital R small r,
Mm-hmm.
then it's capital R small r,
Okay.
then small r small r.
Okay. Now let's go to genotypic ratio.
Genotypic ratio is
How many different genotypes are possible? Tell me. One is this. Is to
then
this.
capital R small r, then small r small r.
Now tell me the ratio.
1:2:1.
Everyone clear? Is this the same like it was in the law of dominance?
No.
Yes.
No, genotypic is correct. And
Yeah,
same.
genotype is same as it was in the law of dominance normally, right? Because genotype only tells us about the alleles. It does not tell us about what happens when those alleles interact. So the difference comes here. If you see phenotypic ratio, now earlier there was only two possibilities, red and white, following the law of Mendel. Now there is a possibility which is red, an intermediate which is pink and white, right?
Yeah.
Tell me how many of them are red?
1.
Only this is red. How many are pink?
2.
And how many are white?
1.
So the genotypic ratio is equal to phenotypic ratio if there is incomplete dominance, because incomplete dominance only affect the phenotype. It does not affect the genotype, right? Write down. Incomplete dominance affects the phenotype, but not the genotype. And in the end, let's define incomplete dominance. So write down.
Sir,
Yes, Arpit.
how it became a ratio to ratio?
Which one? GR or PR?
The phenotypic ratio.
Look here. There was incomplete dominance, right, Harpit?
Yes, sir.
Only two homozygous capital R alleles can give rise to red flower, because this will also produce red pigment. This will also produce red pigment. Red plus red is equal to red.
Right.
Only these two combinations, small r allele, can produce white flowers, white and white. Incomplete dominance means when these two are present as a heterozygous state, this red is not able to dominate on the small r. So there is no complete domination. That's why it's called incomplete dominance. Or in some books, they also write it as partial dominance. Because R is not completely dominant over small r. Hence, this combination cannot give rise to red flowers. Normally, it should have given rise to red flowers, right? But the exception is it's giving rise to pink flowers. Now come back here. Can you see that two of the genotypes are capital R, small r? So they will give rise to pink flowers, right? Because there is law of incomplete dominance in Snapdragon. So the phenotypic ratio becomes 1 red, 2 pink, and 1 white. 1:2:1. Clear?
Yes, sir.
Yes, Arpit? Pakka? Don't just say yes because you have to necessarily understand. You know, when I was studying genetics, I like this subject very much now. It's one of my favorite and strongest subjects. When I studied genetics for the first time, I was like, what is happening? R, small r, so many R's. The reason I started hating maths was they started including alphabets in it. Not hating, hate is a strong word, but not liking maths that much. And the same reason I started disliking genetics in the beginning. And now I did my master's research involving genetics.
I don't
Yes, Father.
like genetics. I was so mad because they included mathematics in it.
Yes, exactly, sir.
Exactly, right. I'm telling you the same thing. No, my disliking for mathematics started because till it was all numbers, I was very happy. Yes, it's happening. You can solve it. Then alphabets came. And I'm like, why? Because, when you are developing as student in school, you have some likenesses, you have some dislikenesses as subjects, and this is how we decide what we want to be, right? Now, I realize that, mathematics is a very important part of even biological research. So we have, whenever we go for master research or studies or PhDs, we are given courses about the mathematics that has to be used for research. Mostly that is your integration, differentiation, and probability and statistics, because you have to give a significant data. Any data in biology, if it is not statistically significant, if it is not backed by mathematics, it is not accepted. Okay. So now story is different, but I can understand why you are not understanding. Okay, Arpit. So there is no need for anyone to feel shy or in a peer pressure has to just say yes if you are not understanding. Ask me 10 times, I will explain you 10 times. We should not move forward until everyone is on the same page, because from here onwards things are going to get more complex. This is not all. This is just we are scratching the tip of the iceberg. That's all. So is it clear, everyone?
Yes, sir.
Okay, very good. So you understood that what is incomplete dominance. Now write down. Let's define it. I have not defined it for boards. We have to write something in board also. Write down. Incomplete dominance is a type of gene interaction where two alleles are interacting. Okay. Incomplete dominance is a type of gene interaction in which both alleles of a gene are partially expressed and in a heterozygous condition are partially expressed, and in a heterozygous condition no one allele is able to exert dominance or show dominance over the other. Full stop. This results in an intermediate or different phenotype. Okay, everyone?
Yes, sir.
Is it clear? Now, they can also ask you the question to make it difficult in entrance or boards, where they say that in a plant, which is not a pea plant, okay, in a plant, there were two alleles for the genes of height, capital T and small t. Now, what will happen if we cross a homozygous or a pure line tall with a pure line short or a homozygous short, given that this species has incomplete dominance in this gene? So you have to be very careful, okay? Don't just see tall and short and just make a cross. Also consider there is an incomplete dominance in the two. So when you make the cross, whenever there is a heterozygous condition, you have to know that phenotypically it means different. So between a tall and a short, if there is a capital T, small t, what will that give rise to? If there is an incomplete dominance?
Medium plant.
Medium height, intermediate height, right? So just be careful about that. Cool. So this was the first exception for law of dominance, which was figured out much after Mendel. There is another exception for law of dominance, which Fatima used very early in this class, known as codominance, right, Fatima?
Yes, sir.
What do we mean by the word co-education, cooperation, anything which is co?
Doing something collectively.
Together, yes. Doing something together. Co-education means co-ed schools are where both the genders study together, right? Then cooperation is like any operation which is done in collaboration or together, which means both the parties are holding weightage. So codominance is also an exception to law of dominance, because in a heterozygous condition, both the alleles are fully expressed. No blending. It's not like giving you example, if there is a allele for red and if there is a allele for white, and if they have incomplete dominance, they will give rise to pink as a capital R, small r. Do you understand? But if there is a codominance, you will see that some patches of the flower will be white, some patches of the flower will be red, some patches will be white, some patches will be red. That is called codominance. You understand? No one is willing to get blend, or no one is willing to give up. Both will express their proteins, and both will express it well and fully. So they are together dominant. So no one is recessive. Do you understand? In a heterozygous condition.
Yes, sir.
And this type of one example for this type of gene interaction is found in human blood group. write down. Codominance is where codominance is a type of gene interaction where two alleles of the same gene, in a heterozygous conditions are expressed together, are expressed separately and equally. Are expressed separately and equally. Okay. Separately and equally. To give rise to different traits in the same individual. To give rise to not a mixed trait, but different traits in the same organism.
To give rise to
Different traits in the same organism. Alleles, heterozygous condition, are equally expressed. Okay. Now this happens in the ABO blood group. Example is AB blood group. You know human blood group, everyone? How many types of blood groups are possible in human? Tell me, how many blood groups you have seen in normal life? What is your blood group, Fatima, if you would like to disclose? You have all the rights not to disclose. You know that? Would you like to tell your blood group so that we can start
A
with
positive.
one?
Mine is A positive.
Your is A positive. So let's keep positive, negative aside because that is because of something called RH factor. That is a different discussion altogether. So let's start with A. So there is A blood group in among us. So do you know, Arpit, the possibility for a blood group genotypically? What are the possibilities? Either you received A allele from the mother and also from your father. So IA is one possibility. Or you received A allele from one parent and received the O allele, or also known as small I allele from the other parent. These are the two possibilities to get A blood group. You understand? Now in this, let me first explain to you. For a blood group, there are three alleles. The first allele is IA. Which gives rise to a sugar which codes for antigen A. Okay.
sir, the I is supposed to be small letter I. No?
No, that small letter I is used for IO. I'm talking about say like alleles. Second is IB, which is for antigen B. The third one is called this small I allele, which is for O, which gives rise to no antigen. So basically this O is nil, zero, but asannata. And it is called an empty allele sometimes, but actually it is an allele. It is a version of the gene which do not give rise to any sugar. So no sugar produced. So if there is no sugar, then no antigen. Because this antigen is a sugar molecule. So understood? So for A blood group, the possibility is IAIA or IAIO. Yes, in this condition, law of dominancy is being followed. Look here, it is a heterozygous condition. Yes, everyone, say yes or no.
Yes.
And this is the dominant allele and this is the recessive allele. Okay. IA always dominates over IO because IO do not produce any sugar. So only IA will be produced and that will show. So basically blood, what is a blood group? Blood group is, you have your RBCs, right? You know RBC? RBC is a biconcave cell, right? On the surface of RBCs, there are some antigens. If it is antigen A, then it is blood group A. If it is antigen B, it is blood group B. If it is, if you have allele O, there is no antigen on the surface. So if someone's blood group is O, which is me, my blood group is O positive, then the RBCs will not have any antigen, not A, not B. Is it clear? It's not like they will have a O antigen. Is this thing clear, everyone?
Yes, sir.
For O blood group, it can be only I and I.
Yes, yes, we're coming to that. So A blood group, these two possibilities are there. Anyone with B blood group in the group?
I'm B positive.
Okay. So for Fatima, let's say what could be the possibilities? Either she received B from both the parents, or she received B from one and a small allele, small I allele from the other, giving rise to this genotype, right? Again, law of dominance is being followed here. If someone is O, I am O. So for O blood group, there is only one possibility. I received small I allele from both my parents. This is the only possibility in a, so because this is a recessive allele. But codominancy comes in this fourth special condition, which is called AB blood group. Have you heard of this blood group? Anyone AB here?
My brothers are
Your brother is?
AB, yeah.
Perfect. So let's study your brother's blood group. What's the reason? He received one A allele from one of your parents and B from another parent. And both of them, this particular blood group shows codominance effect. So both A and B antigens are produced on the surface. So on the surface of the RBC, you will see there will be A as well, there will be B, and both will be present equally everywhere. You understand? This is called a AB blood group, and it has codominance. Is it clear, everyone?
Yes, sir.
So this is also an exception for law of dominance given by Mendel. And what else I want to tell? Yes, we will do some questions on this. The kind of questions that come from this part is what if parents have this blood group, which progeny is possible, which is not possible. If